Hi, I'm Zach Lieber from the Broad Institute, here to present to you on how we use Tableau to track and optimize all of our DNA sequencing operations. So the Broad Institute was founded in 2003 as a nonprofit organization collaboration between Harvard, MIT, and the Broad family. In 2009, we became an independent nonprofit, but continue to operate closely with all of the area hospitals and scientists. So right now you're looking at pictures of our sequencing center, which is the largest sequencing center in the world. And we've been using Tableau since 2010 to optimize our operations. Essentially, we are a DNA sequencing factory. So our input is thousands, really tens of thousands of human and animal samples. And our output are trillions of nucleotides. These are the bases representing the DNA sequences of all living creatures. And these bases are output and used in large scientific studies to uncover the basis for disease. So the mission of the Broad is to reduce disease by coming up with treatments based on the genetic understanding of those diseases. So we are a very high throughput complex operation. We have lots of lab space with robotic machines to handle samples. We have tens of millions of dollars of sequencing machines that process these things thousands of times faster than we were able to do just a few years ago. And then once all that data is made available, we send it off to our compute center, where we have thousands of cores crunching this data for days, if not weeks at a time sometimes, in order to make sense of it all. And we use Tableau to track all of these high throughput operations to make sure that we're getting adequate utilization and yields to make sure that we get the best value for our scientific dollar. So software helps us see the work, and that's really what the approach that we've taken at Broad is, to unravel these complex processes by visualizing them so it becomes obvious to everyone from management to technical staff whether we have a problem or not. So I'm going to walk you through these three applications today that track our lab processing, our DNA sequencing, and our analysis. So let's start with the first one. Our lab workflow, when we receive samples, is pretty long, complex, and branched. It can take several weeks, and depending on the type of samples we receive, whether they be human or non-human, RNA or DNA, these plates of samples, as they move through our lab, will go into various different processes, and they will branch throughout our operation. Additionally, our physical plant is physically disconnected. That is, we've grown up in an urban environment, so we have lab space in multiple buildings that are not connected. So samples may start out in one building and then get transferred to another building for ongoing process. Even once they get to within the same building, depending on the batch type, what you'll see is that the samples will take very different routes, going into different processing centers depending on what steps they need. And if you were to take a closer look at what the processing looked like at any one of those stations, you would have a very difficult time seeing the workflow or seeing the workload. So on the left, you see a picture which was someone's attempt to diagram the workflow as to how samples move throughout our lab. And on the right, you see a picture of basically what a workbench looks like. There are small rectangular plates of samples you may be able to see on the lab bench there that get inserted into these giant robots for processing. But basically, when you go on to our lab floor, there's no way for you to see the flow of work or how much work is piling up, and it's difficult to assess the overall performance of our process. So we developed software to help us see the work. This software overcomes the physical barriers that allow us to visualize the work, allowing us to leverage the human computer, that is, using the human brain to figure out where we've got problems in our system. And the goal of this, and the results have been terrific to date, are to improve our operations. So we are now going to visualize the human operations. So these lab batches consists of plates, which are those rectangular plates there, each of which can contain up to 96 samples in individual tubes. These tubes have purple caps, so you can see them there. And these tubes also have barcodes on them, and the plates that they're inserted into have barcodes on them. And so when these lab batches get to various steps, we will scan them into our database. We actually use a JIRA database, which is an issue tracking system, but we use it to track all of our work. So that in this database, we now know the location of where these samples are. However, it's a very text-based system. There's not a good graphical way of visualizing the results. But that's where we've developed VLAB. VLAB takes all of this data and creates visuals to show you exactly where all the samples are at every step in the process. So arrayed from left to right here is a high-level roll-up of the lab tasks involved in processing DNA. It comes in on the left. That pile is the to-do pile. And the rightmost column there are all those samples that have been done in the last week. Everything in between is in process. The different colors represent different types of DNA. The size of those rectangles is telling us how many samples are in that batch. So the amount of work required to process these samples is a function both of the plate count and the sample count, and we're able to represent both in this display. So at an instant, someone can visualize this and figure out where we've got potential bottlenecks, backlogs, queue problems to deal with, and also recognize their types of samples by quickly looking for the color of samples representing their batch type. One of the features, and we'll go over a lot of the features in the live demo shortly, is to be able to highlight individual batches by age. So we have a dynamic bar chart on the lower right that basically ages all of the samples in process in order for us to be able to highlight both new work that's just come into the system and old work that probably needs to be expedited. Another powerful feature is to be able to split these workflows out. So the previous view is an overview of all the work we're doing at Broad simultaneously. However, as I mentioned before, each of these batch types go through different workflows, and so we can split out different concurrent lanes to track the individual batches as they're moving through their set of human and machine constraints. Each of these product lines essentially has a different target throughput. In this case, you can see a red line on the top, which is a certain target limit that we're trying to make, but we could have different limits for each of these workflows. One of the latest features that we've added is the ability to support a pull system. When we started this project, we were in a push world where we were pushing as much work as possible into the lab when it came to us. We realized this was creating a lot of bottlenecks in the lab, a lot of queues were building up, and it wasn't making anything go much faster. So we switched in the last 6 months to a lean manufacturing approach based on the Toyota method that uses physical Kanban signals in order to tell each step whether more work should or shouldn't be done. In the top of this slide, you can see actual pictures of our controlled refrigerator where we have cardboard boxes that actually tell lab technicians whether they need to be making some more plates or not. And so a full box means don't do any more work, I'm full. An empty box means I need more work. And we're able to overlay those actual box sizes according to our targets directly on the VLAB display so we can see whether we've got the necessary amount of work at each step in order to keep the lines moving smoothly. We also have the ability to assess the total number of samples that are in process for a particular product line and figure out how many samples we were actually able to close out the week before. And in this case, we feel that past performance is the best indicator of future results. That is, if we know that we were able to close out 122 samples last week, and we've got 431 samples in process, that's about a 3.5-week queue time. This is actually based on Little's Law from 1961, but it's serving to be a very accurate representation of what new orders can expect as far as their queue times going through our system. And finally, one of the other features I wanted to point out is that the previous displays I've been showing you are showing you the amount of work stacked up at each step in our lab process as a function of samples and plates, because that's what is required to be worked on in the lab. But once it gets through the processing lab, it then has to be loaded onto our sequencers. We have a fixed number of sequencers, each of which has a fixed capacity. So we want to make sure that we're processing enough DNA every week in order to keep our sequencers full. And that's represented by the green target line that you see on the rightmost column there, as far as how much we want to get done in any given week. So what we've done in this view is we've converted those graphics from, instead of showing a representation of samples and plates, we've actually converted it into the amount of downstream sequencing that's required. Because, as you may see, those types of samples that are purple require a lot more sequencing than those types of samples that are blue, hence the very large purple bars. They require about 10 times as much sequencing per sample, so we need to know not only how much work is that going to be for the lab, but how much is it going to be for the next lab downstream when we try to load these onto the sequencers. Because we don't want to have too much work in progress or too little. We're trying to maximize our available capital. And we think that this feature can also be useful not just for conversion into sequencing capacity, but we could be assessing the value of our work in progress in dollars, the physical volume of our work in progress in numbers of containers that it will take to load onto a train or a ship. And so we're actually positioning this system as a way of tracking any type of complex manufacturing or transportation workflow. So let me take you live into the software for a moment to show you some of the other features and just how interactive it looks. So here is VLAB. VLAB has become the go-to application here at Broad for both figuring out where the work is and explaining to customers how long their samples are going to take. So here you can see a live view. This was updated about 20 minutes ago from our database, showing you every single plate that we've got in process. If we're hovering over one of these, we'll start to see some details, the number of the set, the type it is, and what order this was placed as a result from, and the number of samples in that. And so this is an assessment of how much work is in progress. Our ideal system here would be a lot of evenly spaced bars indicating that work is flowing through the system smoothly. What we don't want is to have huge stacks at any one step. So actually right now our pull system is working very well. We've got a good flow of product through our production line. So we've got a high amount of work done in the last week. We've got a lot of items yet to do, but we're not creating any queues in the middle of the processing, which is very important. The whole approach of a lean manufacturing system is don't pull more work into your system and start adding value to it until you're ready to see it all the way through. It's better to keep things stacked up out here to do. So I mentioned before we've got the age highlighting here, so we can see what are the new samples that have come into the system, what are the ones that have been in for 10 to 20 days, and which are the laggards that someone really needs to take a look at. Why are these taking 60+ days when our target is, say, 30 days for a system? So that's one of the features that we've got. Down here we have the capability to change these graphics from this unit of representation, say, to the gigabase view, which will convert this all into a representation of how many downstream gigabases will be required to sequence this work. And so this way we know that we're not going to have too much work for the next step in our process. So this is the main view that's showing all of our different types of work all grouped together. But we also have several other views. I mentioned before the multi-workflow view. So now we're looking at 3 independent workflows. That is, it's different people, it's different equipment working on these, and we've got different pull targets associated with these different product lines as to how much work we want to be doing per day. And right now it's looking like we're doing very well. That is, each of our 3 main production lines are within the confines of our pull boxes, which are graphically represented here. We never want more than red, and we never want less than green in order to keep things moving smoothly. Here we can see again that by assessing how many samples we were able to finish last week for this product, as well as how many samples we have currently in the queue, we can figure out our estimated queue as a useful way of predicting project schedules for our upstream customers. The way that these workflows are configured is with a setup screen that we've created that allows you to specify which statuses and which types of batches we want to display on each of these pull lanes, as well as a target plates per day, which will set up the size of these pull boxes that we overlay. Another thing that we include in this view is a way of trying to figure out how to set up your workflows. So this example uses parallel workflows, in that our three main product lines are all being done by different people. But you can also use this to set up sequential workflows where all the batches will go from Lab A into Lab B into Lab C, or divergent or convergent workflows. So by using 3 different tracks, which is the number that we chose for our multi-workflow, we're able to track batches of DNA that may have a common set of processes but then split based on batch size. And all of this is useful for figuring out where we've got potential bottlenecks in the system. One of the very latest features that we've added, just last week in fact, was the ability to figure out how this lab work and lab batches map to customer orders. So each of these batches are approximately 100 samples. Some are larger, some are fewer. But the customer orders come in, and the customer orders may be for 10 samples, or they may be for 1,000 samples. If we get a bunch of orders in for 10 samples, they'll all be grouped together and put onto one batch. If we get orders for 1,000 samples, we'll have to split them up and put them in 10 different batches. So this allows us to say, look at the status of some of our larger projects and see how those batches are moving throughout the lab. So we have an overall feel for when this project might be done, how much data will be available soon, and whether any of the samples in this particular order might be stuck here in this on-hold column. So that's one type of order. And then here's another order, for example, where we can see a different type of batch moving through the system, but a way of seeing how that overall order is being distributed throughout the lab. So these are all features that we're adding in order to get a visual representation of where is the work in process, where are the customer orders, do we have enough work in the lab, and are we going to be able to keep the sequencers busy next week. That's our timeline. That is, this is about a 2 or 3 week lab process, and then it goes into about a week of sequencing and a week of data analysis to close it out. So what we've done, if you imagine, is we've taken this complex workflow that branches throughout our multiple buildings, and there's no way to assess how much work is actually in flight at a given time. And we've converted that into a very nice visual view of that that gives us an exact indication of where all of our batches are at any given time, how long they're going to take to get through the system, how long they've been in the system, and whether that's an adequate amount of work. So what we call this is VLAB, and what VLAB does is it lets us see through walls. That is, we do not have the ability to have a large manufacturing floor that lets us physically observe all of this work in process. Because we've grown up in an urban environment with vertical buildings, we have to use software to overcome those physical barriers, let us see the work, and then once our managers and technicians can see the work, they know how to make a huge improvement on it. So that's been one of the applications that we've developed in Tableau over the last 6 months. And it's been widely adopted and is now the standard that we use at Broad for tracking and optimizing our samples. The next thing I'd like to show you is a couple of our other applications. I think I can talk through some of them actually. I mentioned that first we do lab processing. The next step is to load all these samples onto our sequencers. Our sequencers cost $1 million each. We have 50 of them. We turn them over every few years. It's a very expensive, capital-intensive operation. We need to make sure that we're utilizing those maximally. So we have different models of sequencers, some of which run in one day, and some of which take 1 week to run, depending on whether you're going for high output or high speed. And we're tracking all of those together in this display, where the leftmost pane, you're seeing the start and end times for each of the runs. The ones on the top are week-long, the ones at the bottom are day-long. Then we're seeing what percentage utilization each of those machines has over a 24-hour period, or 168 hours in a week. We're seeing exactly how many hours that machine was in use versus not in use to give us an average utilization rate, which we're trying to get up to, say, 80%. Then the output is shown in column 3. So the output of each of those runs in billions of bases is shown by those shown by the colored circles. And so the machines that run for a week have higher output, obviously, than the machines that run for a day and have lower output. But what really matters is the total amount of bases that these machines can put out in a given week, whether they're running high speed or low speed. And so this is how we're tracking what our average, minimum, and maximum output is at each of our machines to look for trouble where a machine might need to be serviced, or to gain confidence in what a steady state output will be so that we can do planning, setting our targets for how many bases we can actually sequence the following week. And then those targets are fed upstream into VLAB in order to tell us how much work we should be doing. So that was an instantaneous view of all the work that had been done recently. We also have a view that tracks this over time. So we can go back in history, figure out how many machines we had in the building that were operational at a given time, and then figure out what percentage of those were being utilized at any given day. And we found something really interesting out when we first started doing this for our high-speed machines, which are shown there on the bottom. So traditionally we'd use the machines on the top that took 1 week to run. Then we switched to these machines on the bottom that took 1 day to run. And what we realized was that because we didn't have a weekend shift coming in, these machines would get loaded up on Friday, they'd be finished on Saturday, and they'd sit idle on Sunday. And that's why exactly once a week you see the utilization rate drop to 0 for these new high-speed machines. Essentially, they became too much work for us to keep loaded until we trained a weekend shift, which now you can see we did around August. So now we no longer have our weekend dropouts, and we're now getting our sustained utilization up to about 80% of theoretical capacity for these high-speed machines. And it just became very obvious to us visually that we needed to hire a third shift because of these dropouts depressing our average rate. So even though when we were in the building it seemed like we were really busy, when you look at it with data, you realize that 1 or 2 days of downtime was really hurting our overall utilization. To do this type of work where we're converting run start and end times into number of runs in process at any given time in the past requires a little bit of data reshaping. And it's very useful to have this in your arsenal, whether you're trying to track machine utilization or number of open orders or number of open help tickets. This exact same algorithm works in all of those cases. So what you do is you will transform a set of data that's got 1,000 rows, each of which has a start and an end date, into a set of data with 2,000 rows, each of with a date, either incrementing or decrementing the number of active jobs we have. So in this case, we're selecting our start date as the date and adding 1 to the number of flow cells that are running, and we're unioning that with all of the run end dates, each of which is associated with a -1. So then all you have to do is put that into Tableau and do a continuous running sum over date, and you'll know exactly how many flow cells, jobs, tickets, etc. were open at any given date in the past. And so we use this to convert all of our open tickets into open queues throughout the lab. Finally, the last step in our process is to send all of this data off the sequencers and send it into our compute farm or our cloud-based compute in order to analyze this data. And these projects have thousands of samples, and these samples are split across dozens of flow cells, run across dozens of different machines, and then the algorithms will link all of this data back up together at the end. But it's very hard to keep track of exactly what's happened lately on your project. So we developed this application called SeqMinder, which will show you along a date axis everything that's come off of our analysis pipeline is shown by a graphical mark here, either a square or a plus. The colors are coded based on whether those samples have been billed or not yet. And these are the different projects and orders that we have. And then by selecting any one of these marks, what we do is we get a coverage chart that's saying, for this order, it looks like it contains 398 samples. This is showing the progress of each of those samples. So everything marked in green are those samples that have met their target coverage. Those samples in orange haven't quite met their target coverage, and those samples that are in red haven't even made it onto the sequencer yet. So this is showing a project that's in process. And this is the only way of really tracking large-scale projects, particularly when, as a project manager, you may be tracking dozens of large-scale projects. It's hard to check in on every one. What you really want to do is just put a list of all these projects into SeqMinder and then look for anything new coming off the right side of the chart, indicating you've got a new flow cell run or a new analysis run. So this type of timeline interaction with the ability to select marks and see the overall progress of your project has also been very helpful. So by using VLAB to check our lab work, the HiSeq Dashboard to check the status of our sequencers, and SeqMinder to check the status of analysis, we've got a visual on how to keep track of our high throughput, very complex operations. So we do everything with Tableau at Broad. This was just a sample of the type of stuff that we do. We're tracking yields, we're looking at service downtimes here, we're tracking queues. We even have reports that are tracking exactly which reports on our Tableau server all of our users are using to see which reports are hot and which reports are not. The results there are quite surprising to see reports that we didn't think people should be using and they're using a lot, and the ones we think they should be using, they're not. And so we use this to steer the way we market new reports to our user base. We have about 200 users at Broad of Tableau, about 60 of which log in on any given weekday. That's shown in the lower right quadrant there. Those blue bars are showing you the weekdays and then the weekend in between. So we track everything from finances to utilization to user use in this. So if I could give you any message about how you can improve your operations with analytics, the first step is that you have to collect process data. You have to figure out how to instrument the key processes in your environment to collect some type of data that can be loaded in, even if it only goes into a spreadsheet, preferably a database. Then you have to take that raw data and transform it into useful metrics. So we didn't have a bit of raw data that showed us what our utilization was. We had to derive utilization by transforming the raw data. So figure out what a useful metric is, figure out what data you have available to you, and then figure out how you can get that data into something more useful. And then make sure you present that to your users clearly and with context. By context, I mean either more detail or a historical timeline to show how the current performance relates to past performance. And we found that just by using clear presentations in the right lab and management review meetings, everyone gets on board with honing in on exactly what our major problems are, and that visibility is what's enabled us to run such an efficient operation. So we're very pleased to be Tableau users and are looking forward to extending this type of analytics throughout other parts of Broad. So we've started in the Genomics Platform doing this, but are also working with departments throughout the Broad and to bring this type of analysis to their operations as well. Thank you very much, and I hope you enjoyed this presentation.